We now move into a class of lipids that I like to call structural lipids. And the most important member of this class are the phospholipids, which are the chief structural component of cell membranes. I'm sure in a biology class or biology book you have seen pictures of the plasma membrane of cells, and you probably see a circle on the top and a line or maybe two lines coming off of it, and you have two of these together, and that causes the lipid bilayer characteristic of cell membranes. Well, let's look in a little more detail from an organic chemist perspective at what the structure of these phospholipids actually is. So just like triglycerides, they have glycerol as their core, except instead of being triesters of glycerol, they are diesters of glycerol. So there are two fatty acid molecules. Those are the two lines that come down off of the head of the phospholipid. But then the third OH of glycerol is linked to a phosphate ester. Okay, so a phosphorus and then an OR group. So if R is hydrogen, the parent phosphoric acid is called phosphatidic acid, and that's going to help us understand the names of some of the molecules that we're going to meet in this lesson. So two common R groups that are present are ethanolamine and choline, both reasonably abundant small molecules in the body. And the resulting phospholipids that you get from using these R groups are phosphatidylethanolamine, which is also called cephalin, and phosphatidylcholine, which is also called lecithin. Two other important phospholipids are phosphatidylserine and phosphatidylinositol. Okay, so serine is one of the 20 naturally occurring amino acids. It has an OH on it coming off of the side chain, and that OH can become bound to the phosphate. So I have the different pieces of this molecule color-coded. So in green are the fatty acid pieces, in blue are the glycerol pieces, black is the phosphate, and then red is the alcohol fragment that forms the phosphate ester, which in this case is serine. Another important one is phosphatidylinositol. Again, green, blue, and black pieces are the same, but now instead of the amino acid serine, the alcohol piece is actually a molecule that is called myoinositol, which is cyclohexane with 6 OHs, so there's an OH on every carbon, and the stereochemistry shown, so having 2 of them up and the other 4 down, is why it's called myoinositol. That's the most common form of inositol. But there are actually other inositols out there with different relative stereochemistries. So inositol, if you care, is actually biosynthesized from glucose, having 6 carbons and an oxygen on each carbon. So having moved from basic phospholipids, we now need to look at what are called sphingolipids. And those are phospholipids that are based on a sphingosine backbone in blue rather than glycerol. So drawn on the screen here is the structure of sphingosine. You can't really appreciate from the way I drew it exactly how large this structure is, because it has a C13H27 chain, so just all carbons and hydrogens coming off of the left-hand end of it. So it itself is a molecule that has a polar head, 2 OHs and a charged NH3, and a nonpolar tail. These are linked by a trans double bond. So the most common sphingolipid is what's called sphingomyelin, and that's a component of many cell membranes, but the name should ring a bell to you. It's actually abundant in the myelin sheath of nerve cells. So once again, the color coding is the same. The sphingosine, it now is in blue. The fatty acid is in green. And you should be able to see here that the fatty acid actually forms an amide linkage to sphingosine rather than an ester linkage. It could have formed an ester. There is an OH available to it, but for whatever reason, the linkage is an amide linkage. And then the phosphate is in black. And then finally, the piece in red, the alcohol group, is actually a choline, which we've already seen in phosphatidylcholine. So another class of lipids that you should be aware of are the glycolipids, and those are ones that have a sugar in the place of the phosphate ester. Okay, so you can have a glycerol-based glycolipid, so this looks exactly like a phospholipid. 2 of the 3 oxygens of glycerol are esters with fatty acids, but then instead of having the phosphate ester, you have, for example, here is a molecule of glucose, and there's an alpha glycoside bond present there. Or you could have a sphingoglycolipid, that's turning into a tongue twister, I know that, where you have the sphingosine fragment, and it's also using its terminal alcohol to become bound to a molecule of glucose. So glycolipids generally exist on the surface of cells and are involved in cell signaling. Okay, so they are recognized, they're sometimes receptors, and they're recognized by small molecules. So the last structural lipid are waxes, and I know what you're thinking. Earwax, right? It's definitely a kind of wax, and one that I generally consider to be quite annoying. But what is a wax? Well, waxes are high molecular weight esters, high enough molecular weight with enough carbon atoms that they are going to be solids. Okay, so drawn on the screen here is the structure of triacontyl hexadecanoate. Say that 5 times fast, that's another tongue twister for you. But if you remember from the nomenclature of esters, what's to the right of the oxygen in the chain comes first, and that's a 30 carbon chain, thus the tri in the name of it. And then on the left-hand side it's a derivative of hexadecanoic acid of a 16 carbon carboxylic acid. And this turns out to be a major component of beeswax. Okay, so that's a more useful type of ester. And then I just want to leave you with some other biological functions of waxes. So, sperm whales use waxes to detect sound waves. So, whales can map out their environment using sonar. And then waxes also tend to be found in bird feathers, mammalian fur, and plant leaves. So they have, many of them have waxes coating their surfaces, things that live outside in order to be able to repel water when it rains, or in case of certain plants, be able to keep the water that they need inside the plant pot. Okay, so structural lipids, phospholipids, glycolipids, sphingolipids, and waxes. So our next stop in the tour through lipids is going to be the steroids.